Okay, let's talk about genetic modification quickly. So, genetic modification is when you add or remove genes from an organism, okay? And if you've added genes or genetic material from a different organism, we now call the resulting organism that you've modified, we call it transgenic, okay? So, transgenic organisms. So, if we think about the example of insulin, so type 1 diabetics, they do not produce insulin, they need to inject it. That means that we need to have insulin in large doses, human insulin. So, what we could do is we can get some cells from a healthy patient and extract their DNA. Not patient, sorry, a healthy individual, extract their DNA. Now, their DNA will have a gene for insulin, right? And we know what gene codes for or produces, insulin. So we isolate that gene, we find it, and once we found it, we use an enzyme to cut it out. We call that enzyme a restriction enzyme. So we cut out the gene we want, and let's say this is the gene over here, okay? The one that makes insulin. Now, the restriction enzyme, when we essentially cut it out, kind of like chemical scissors, it leaves sort of a jagged end, not a smooth end. And we call this end a sticky end because it can form, it's not all the bases are paired over here. So the bases can pair again. Let me show you on a different diagram that I have. So you see why these are sticky ends? Because those two C's can still pair with two G's, right? It's not complete. Likewise, these two G's could pair with two C's, right? So they're sticky ends. But we are only going to keep one of this. So one of these is the human insulin gene, okay? So human insulin gene, once we have it, we kind of put it to one side. Then we turn our attention to bacterial cells. Now, bacteria divide really, really quickly. They reproduce quickly by binary fission, so they are useful to us. They also have plasmids, which you know from Unit 1. These plasmids are extra bits of DNA, and the great thing is you can take a plasmid out of a bacterial cell without killing the bacterial cell, and you can reinsert it. So what we do is we take out a plasmid, okay, and we select an area about the same size as our insulin gene, and we cut it out to get rid of it. We don't need it, okay? And we're going to cut it out with the same restriction enzyme that we used previously. What this will do is it will mean that the empty space that you get here in this plasmid after cutting it out will be complementary to this sticky end. It will match up nicely. You see how this shape fits the human insulin gene that we cut out previously? Sorry, my drawing leaves a lot to be desired, but okay, you see how those two shapes fit nicely together? That is because the sticky ends were complementary. Okay? Now, just like we used an enzyme like complementary, just like we used an enzyme to cut it out, almost like scissors, like chemical scissors, we need an enzyme to join these two together. They're not just going to stick together by themselves. So we join them using an enzyme called DNA ligase. Okay, DNA ligase. So in an exam question, if you're explaining genetic modification, the words that you absolutely have to use: restriction enzyme. Okay, number one, restriction enzyme. Number two, this idea that we use the same restriction enzyme for the bacteria. Okay, number three, so maybe I should number them. Number one, this is the restriction enzyme to cut out gene of interest. Number two, use the same restriction enzyme on plasmid. Okay. Number three, the sticky ends then will be complementary to each other. Okay, these sticky ends. Number four, join them. Usually, even the idea of joining or inserting can be a mark, but you need to specifically say DNA ligase. So join them, DNA ligase. Sorry, it's not usually. I was saying that potentially you could get a mark for joining them, but most of the time you need to mention DNA ligase. Okay. Now, then this would be 4 marks. If the question was any longer than 4 marks, you would then need to say, step 5, you're going to reinsert plasmid, so the modified plasmid, this is the modified one, the transgenic one, into the bacterial cell. Now, why this is useful, they'll take this bacterial cell or these bacterial cells, they'll put them into a a huge vat, which is a long container with the right temperature, the right amount of nutrients. And basically, what that will do is it will give the bacteria the right conditions it needs to divide. It will make loads and loads of copies of each other and loads of copies of this plasmid, which has the human insulin gene in it, which means that all of those bacterial cells will be making human insulin. We can then purify that insulin, meaning we can get rid of the bacteria from it, and voila, we have pure human insulin. I hope that made sense. Come and see me if it doesn't.